Next story. Unlocking the role of microbiome in fighting malnutrition. In the quest to combat malnutrition, a groundbreaking study published in Nature Microbiology by Chang et al. Reveals a novel approach to establish causality between the gut microbiome, dietary components, and host physiology. Unraveling the intricate interplay between diet and the gut microbiome is crucial for human health, ranging from metabolic disorders to malnutrition. While human studies can only establish correlations, a reverse translation strategy using GNO to biotic mice has enabled researchers to delve into cause and effect relationships that were previously challenging to ascertain in human trials. The study, spearheaded by a team of scientists, initially conducted a nutritional trial using a microbiota-directed complementary food, MDCF2, to address malnutrition in Bangladeshi children. The results showed significant improvements in weight gain, particularly associated with the abundance of specific Prevotella copri strains in the gut microbiota. Further analysis identified that these strains utilized carbohydrates present in MDCF2, linking their presence to enhanced weight gain in malnourished children. To establish the causal role of P. copri in mediating the effects of MDCF2, the researchers turned to GNO to biotic mice colonized with defined bacterial consortia. Through a meticulously designed experiment, they demonstrated that P. copri strains metabolizing MDCF2 glycans were pivotal in promoting weight gain and modulating energy metabolism in intestinal epithelial cells. Interestingly, the collaboration with Bifidobacterium longum subspecies infantis was crucial in enhancing the colonization of P. copri and optimizing nutrient uptake and metabolism within the host. Despite encountering challenges, such as the inability to monocolonize mice with P. copri strains, the study provides valuable insights into the intricate web of interactions within the gut microbiome. By shedding light on the mechanistic pathways through which specific bacterial strains influence host physiology, this research sets the stage for developing targeted nutritional interventions for malnourished children. Moreover, the reverse translation strategy holds promise for unraveling the nuanced connections between diet, the microbiome, and human health in broader contexts beyond malnutrition. This groundbreaking study not only underscores the importance of exploring microbiome causality but also highlights the potential of leveraging animal models to uncover hidden mechanisms that shape our health outcomes. Moving forward, such innovative approaches pave the way for designing tailored dietary interventions that harness the power of the microbiome to promote human well-being. Source: Nature Microbiology, Nature. Next story. Unraveling the mystery of quivering eyes and chemistry woes. In a thrilling journey through the annals of scientific history, we uncover fascinating insights into the complexities of human vision and the ever-expanding realm of chemistry. Delving into the past, we encounter the work of R.W. Ditchburn, who shed light on the phenomenon of constantly quivering eyes. Despite our perception of stillness during focused gaze, our eyes are in a continuous dance of tremors, drifts, and flicks that are essential for maintaining our perception of the visual world. This discovery not only highlights the intricate workings of our visual system but also underscores the dynamic nature of our sensory experiences. Moving back even further, we are transported 150 years ago when chemistry was described as a science so vast that its handbook required a wheelbarrow for transport. The limitless capacity for development within the realm of chemistry poses a unique challenge for scientific inquiry, with constant additions to knowledge making a comprehensive understanding increasingly elusive. As the science of chemistry encompasses the entirety of nature, from animate to inanimate, its breadth and depth present a formidable yet enticing field for exploration and discovery. These snippets from the past serve as a reminder of the enduring curiosity and relentless pursuit of knowledge that drive scientific endeavors. From the subtle movements of our eyes shaping our perception of the world to the boundless expanse of chemistry waiting to be unraveled, the tapestry of science is woven with threads of complexity and wonder. As we stand on the shoulders of giants who have explored these realms before us, we are beckoned to continue the journey of discovery, pushing the boundaries of our understanding and expanding the horizons of human knowledge. Join us in this riveting exploration of scientific history, where each discovery and revelation paints a vivid picture of the intricate tapestry of the natural world and the human quest for understanding. Source: Nature. Next story. Unlocking the evolution of bird flight. Celebrating 50 years of groundbreaking research, in the realm of evolutionary biology, there exists a long-standing mystery shrouded in the origins of bird flight. For decades, Scientists grappled with the enigma of whether avian flight emerged from creatures gliding down from trees or from animals running and flapping their way up from the ground. However, a seismic shift occurred 50 years ago with the publication of a seminal paper that forever altered the trajectory of this debate. In 1974, a maverick paleontologist named John Ostrom penned a groundbreaking paper in the Quarterly Review of Biology that upended conventional wisdom and redirected the focus towards the evolution of the flight stroke itself. Ostrom's work was a game-changer, drawing on evidence from both the fossil record and the behavior of extant animals to advocate for a new perspective on the origins of bird flight. Prior to Ostrom's intervention, the origin of bird flight was shrouded in ambiguity, with theories ranging from avian ancestors descending from reptiles to the improbable notion of creatures evolving flight capabilities through tree-dwelling acrobatics. Ostrom, however, boldly proposed that birds evolved from small theropod dinosaurs and that flight evolved from the ground up rather than from the trees down. His meticulous study of Archaeopteryx, the iconic fossil species, alongside his findings on theropod dinosaurs like Deinonychus, unveiled a striking array of similarities that pointed to a direct link between dinosaurs and birds. Ostrom's work elucidated the evolutionary trajectory of flight stroke development, emphasizing the importance of bipedality and cursoriality in laying the foundation for avian flight. Furthermore, Ostrom's insect net hypothesis challenged traditional notions of flight inception, positing that early protobirds may have utilized rudimentary feathers to trap prey, thereby kickstarting the evolution of powered flight. While initially met with skepticism, his ideas paved the way for a deeper understanding of how flight mechanics evolved across various flying vertebrates. By refocusing the debate on the biomechanics of flight, Ostrom's 1974 paper sparked a paradigm shift in evolutionary biology. His insights not only revolutionized our understanding of bird flight origins but also laid the groundwork for future research into the evolution of flight across diverse animal groups. Today, as we commemorate the 50th anniversary of Ostrom's pivotal paper, we continue to unravel the mysteries of avian evolution and flight mechanics. Ostrom's legacy endures as a beacon of scientific inquiry, challenging us to soar to new heights in our quest to comprehend the complexities of the natural world. Source: Nature article, Nature, next story. Addressing bias in AI image generators, can racist and sexist outputs be corrected? In the realm of artificial intelligence, a disturbing trend has emerged with image-generating programs yielding racist and sexist outcomes. The situation was brought to light by Pratyusha Ria-Kalluri, a graduate student at Stanford University, who discovered biases in popular AI tools such as Stable Diffusion and DALL-E. These tools often perpetuated stereotypes, associating poverty with Africa or specific jobs with certain genders and races. The issue with biased AI-generated images stems from the training datasets containing skewed representations of gender, race, and occupations. As a result, these biases are reflected in the images produced by the AI models. The implications of such biased imagery are far-reaching, affecting media, global health organizations, and internet databases. Furthermore, the increasing reliance on AI-generated images could exacerbate societal stereotypes if left unchecked. Efforts to rectify these biases include refining prompts given to AI systems, incorporating diverse images in training data, and enhancing curation processes. However, these solutions bring their own set of challenges. For instance, attempts to counter biases through explicit counterprompts have sometimes backfired, exacerbating existing prejudices. Moreover, the onus is unfairly placed on users, especially those underrepresented in datasets, to mitigate biases in AI-generated images. The need for transparency in AI systems is crucial to understanding and addressing biases effectively. Open sourcing AI models and datasets would enable researchers to identify and rectify sources of bias. Standardized practices for annotating datasets and stringent regulations, like the European Union's AI Act, are essential steps towards ensuring accountability and fairness in AI technologies. As the debate on addressing bias in AI-generated images continues, the ultimate question remains: what should the ideal output of AI reflect? Striking a balance between reflecting reality and promoting fairness poses a complex challenge for researchers and policymakers alike. In the quest for unbiased AI technologies, the journey towards transparency and ethical AI practices is just beginning. Source: Nature. Next story. Assessing the value of the Mars rover's $11 billion rock collection, in the realm of space exploration, the stakes and possibilities have never been higher. As the Perseverance rover on Mars continues its groundbreaking mission, scientists are faced with pivotal decisions that could determine the trajectory of future discoveries. The rover, which has been diligently collecting rock samples from the ancient river delta in Jezero crater, holds within its grasp the potential to unravel the mysteries of Mars's past. With its payload of 17 tubes filled with Martian rock, dirt, and air, the Perseverance rover has amassed a geological treasure trove that could hold the key to unlocking secrets about the red planet's history. The tantalizing prospect of finding signs of past life on Mars looms large, but the crucial next step involves bringing these samples back to Earth for in-depth analysis. The challenge, however, lies in the staggering cost of such a mission, a whopping $11 billion, according to an independent panel's analysis. As NASA and the European Space Agency, ESA, grapple with budget constraints, the fate of the meticulously collected rock samples hangs in the balance. Scientists are keeping a watchful eye on developments, anxiously awaiting a decision on the rover's final destination for sample collection and the feasibility of transporting these precious specimens back to Earth. Amidst the financial dilemma, there is also a spirited debate among Perseverance's planners regarding the rover's next moves. While some advocate for sticking to the original mission plan and exploring the ancient river delta to its fullest extent, others propose a more cautious approach, suggesting a safer and potentially more cost-effective strategy by revisiting lower-altitude sites within the crater. The urgency of these decisions was underscored at the recent Lunar and Planetary Science Conference, where scientists emphasized the transformative potential of the Mars rock samples. Comparisons were drawn to the iconic moon rocks collected during the Apollo missions, hinting at the groundbreaking discoveries that could await within the Martian specimens. As the scientific community holds its breath, NASA faces a critical juncture in the Mars sample return mission. The intricate dance of science, budgetary constraints, and technological challenges will ultimately shape the future of Mars exploration. The dream of bringing Martian rocks back to Earth may hang in the balance, but the quest for knowledge and discovery knows no bounds. In a saga that spans decades of innovation and exploration, the Mars rover's rock collection stands as a testament to human ingenuity and curiosity. Whether the $11 billion price tag proves insurmountable or a pathway is found to overcome financial barriers, the quest to uncover the secrets of Mars remains an enduring testament to humanity's insatiable thirst for knowledge beyond the stars. Source: Nature. Next story. Revolutionizing science. Creating antibodies from scratch with AI. In a groundbreaking discovery that could revolutionize the field of antibody design, scientists have successfully utilized artificial intelligence, AI, to create antibodies from scratch for the very first time. This remarkable achievement, documented in a recent preprint on bioRxiv, could potentially streamline the process of developing therapeutic antibodies and make it more accessible to researchers worldwide. Traditionally, the production of antibodies involved labor-intensive methods such as animal immunization or screening numerous molecules. However, by harnessing the power of AI, researchers have now demonstrated the ability to design novel antibodies with unprecedented efficiency and precision. The AI tool employed in this study, known as RF diffusion, was originally developed by a team at the University of Washington and has been instrumental in advancing protein design. By adapting RF diffusion and training it on a vast dataset of antibody structures, including those involved in crucial interactions with pathogens and disease targets, the researchers were able to generate thousands of antibody designs tailored to specific bacterial, viral, and cancer-related proteins. Upon experimental validation, approximately 1% of the AI-designed antibodies exhibited the desired binding characteristics, showcasing the tool's potential for creating functional antibodies. Despite the initial success, there are still significant challenges to overcome before AI-generated antibodies can be used in clinical settings. Notably, the antibodies generated through this AI approach are categorized as single-domain antibodies, akin to those found in certain animals like camels and sharks. While these antibodies are simpler to design and study in the laboratory, further modifications are necessary to align their sequences with human antibodies for therapeutic purposes. Although the road to developing AI-designed antibodies for clinical use may be long, researchers are optimistic about the prospects. This achievement signifies a pivotal moment in the realm of antibody design, offering a glimpse into a future where cutting-edge technologies could expedite the creation of tailored therapeutics at an unprecedented pace. Source: Nature article. Nature.